To Lecture 12, where we will go over the diversity, structure, and function of inverts and vertebrates. This diagram represents the current phylogeny of the various vertebrates and invertebrate species. Note that, just like we've seen in protists, invertebrates show up in many lineages. Invertebrates are included in Porifora, phylum Cnidaria, the Lophotrochozoans, and the Ecdysozoans. Also note that invertebrate species are present in Deuterostomia. However, the grouping Deuterostomia also includes vertebrate species. Also note the important derived traits that exist, such as the development of tissues in the eumetazoans and the development of bilateral symmetry and 3 tissue layers that are present in the bilaterians. Invertebrates are characterized as animals that lack a backbone. They show a great amount of diversity in both their morphology and their ability to inhabit nearly every single habitat present on Earth today. Invertebrates, as stated previously, are present in many different lineages. The Poriforans, Cnidarians, Lophotrochozoans, and Ecdyozoans all contain invertebrate species, and the deuterostomes contain invertebrates as well as vertebrates. We're first going to take a look at characteristics of the phylum Porifera, or the sponges. Sponges are considered to be a basal lineage that diverged very early from all the rest of the animal phyla. Sponges in general are sedentary filter feeders. They can remove food particles from the surrounding water as the water passes through their body. Water will pass through body pores and enter into the central cavity called the spongiocele. It will then exit out the central pore, which is called the osculum. In general, sponges contain 2 layers of cells that are separated by what is termed the mesohyle. Both layers of cells are in constant contact with water. Thus, these cells can simply use diffusion to carry out gas exchange and to remove waste. Some more complex sponges may have folding to their body layers, water canals, and they may have multiple oscula. This diagram gives you a general overview of the structure of a sponge. Note that the inner layer of cells contains the choanocytes. Also note their similarity to the choanoflagellates. From there is the mesohyle layer, which is then surrounded by the epidermis layer. Also note the presence of pores that will allow for the water flow to flow into the internal chamber. Water will then exit out the top of the animal at the osculum. Although sponges lack true tissues, their body does contain 2 different major cell types. Choanocytes, which resemble the choanoflagellates. These are the cells that were depicted in the picture previous that lines the interior surface of the spongiocele, or the internal cavity. These cells have finger-like projections surrounding a flagellum, which forms a collar. These cells are important in engulfing bacteria and any food particles through the process of phagocytosis. Amoebocytes, on the other hand, are allowed to move throughout the mesohyle. They do this through the use of pseudopodia. Amoebocytes can also consume and carry nutrients to other cells within the sponge. Many amoebocytes can produce sharp skeletal fibers called spicules, or flexible fibers called spongin. Amoebocytes are considered to be totipotent. This means they can retain the ability to become any other cell type. This means that they can undergo differentiation to form an epidermal cell as well as a choanocyte. In general, most sponges are considered to be hermaphroditic. This means that each individual can produce both sperm and eggs. Most are sequential and function as one sex before they change to another. Sperm is usually released generally into the water current, where it can then cross-fertilize with any neighboring females. Zygotes that form from the fusion of the sperm and the egg develop into a flagellated larvae called the amphiblastula. This larvae is important in providing a dispersal mechanism from the parent plant. These amphiblastulas can then swim around and settle on a suitable substrate. Once they settle, they can deposit and become a sessile adult. We're now going to take a look at the next major group of invertebrates, the cnidarians. Cnidarians are considered to have true tissues. They are diploblastic, meaning that they have both ectoderm and endoderm. They also have radial symmetry. Cnidarians have a gastrovascular cavity with a single opening that functions as both the mouth and the anus. Common cnidarians include the hydra, corals, as well as jellyfish. There are two main body forms in cnidarians: the polyp form, which is common in more sessile animals, and the medusa form, which is common in more mobile animals. In the polyp form, the tentacle and mouth face upward. They will adhere to the substrate at the arboreal end or basal end. However, even though they are considered to be sessile, they may be capable of limited movement across the substrate. The polyp body form is very common in hydra and sea anemones. The medusa form, however, has the tentacles and mouth facing downward. These animals can move through their environment through drifting and also through body contraction. This body form is more common in jellies or jellyfish. In general, cnidarians are considered to be predators that use tentacles to bring their food into their mouth and into the gastrovascular cavity, where it can then be digested. Cells that line the gastrovascular cavity can secrete specific enzymes that are important in breaking down the food and also in absorbing the nutrients. Cnidocytes are specialized cells that line the tentacles. These are important cells that will aid in defense mechanisms as well as prey capture. The cnidocytes contain capsules called cnidae. Some of these cells are specialized into nematocysts. These have the ability to pierce and sting prey. However, other cells may be thread-like or adhesive to entangle prey. This diagram depicts the importance of cnidocytes in prey capture in the hydra. At the end of the hydra, at the tentacles, you can see the presence of many or multiple cnidocytes. These cnidocytes have a specialized trigger mechanism. When the trigger is stimulated either by pressure or chemical changes in the environment, the thread, which is present within the nematocyst, will then burst forward. This will then release the toxic contents of the nematocyst that will then help to immobilize prey. Many cnidarians possess simple contractile tissues that are formed from bundles of microfilaments that are arranged parallel in fibers. These fibers can be found between the epidermis and the gastrodermis. In this instance, the gastrovascular cavity can also act as a hydrostatic skeleton, offering support and providing movement for the animal. When the mouth is closed, the organism can change shape as these cells contract. In addition, cnidarians possess a simple nerve net that extends throughout the body. This is important for allowing the organisms to detect and respond to stimuli in their environment. In general, the cnidarians are divided into two major clades: the medusozoans as well as the anthozoans. The medusozoans includes the jellyfish, sea wasps, and hydras. Most of these animals are strictly medusoid in their body form. However, some may alternate between the medusoid and polyp form. This diagram depicts the typical life cycle of a hydrozoan. Note that the reproductive form exists in the medusoid body plan. The medusa will undergo meiosis in the gonadal tissue to eventually form the egg or the sperm. These will join and undergo fertilization to form a zygote. The zygote will develop into a larvae, and the larvae will exist in the polyp form. The mature polyp will eventually develop, and this can eventually form a colony. Note that these colonies are primarily for feeding. However, some polyps will be produced that will end up being a reproductive polyp. In this instance, these would give rise to the medusoid form. The next clade of cnidarians is the anthozoans. These include the sea anemones as well as corals. These organisms are strictly found in the polyp body plan. They can be solitary or colonial, and many of these are symbiotic with different algal species. Many of these individuals can secrete an exoskeleton. However, this exoskeleton is not typical of what we see in other arthropods, for instance. In anthozoans, this exoskeleton instead is made of calcium carbonate. This is important in helping to anchor the base of the polyps within a colony, and it also provides a scaffold or lattice structure for future generations to use. We're now going to take a look at the bilaterians, specifically the Lophotrochozoans. Note the phylogenetic tree that depicts all of the bilaterians. Bilaterians include the Lophotrochozoans, ecdysozoans, as well as the Deuterostomia. Members of the bilaterian group are all triploblastic. They also all exhibit bilateral symmetry. However, despite this, they show a great diversity in their body form. With respect to the Lophotrochozoans, a unique characteristic of these groups include the development of a lophophore or ciliated ring for feeding purposes, and these individuals may also have a trochophore larval stage, which is unique to this group. However, this may not be present in all members of the Lophotrochozoans. The Lophotrochozoans include the Platyhelminthes. These are the flatworms. Flatworms can generally be found in all marine and aquatic habitats. They can be free-living in the environment, or some of them may be parasitic, such as tapeworms and trematodes. Platyhelminthes organisms are all triploblastic, however, they do not have a true coelom and are considered to be a coelomate. They do, however, have ganglia as well as a ventral nerve cord. Their overall flattened shape helps to increase the surface area and also to ensure that all body cells are in contact with the environment so that they can carry out processes such as gas exchange, waste elimination, and osmoregulation. This diagram depicts the overall organization of a flatworm. Note the presence of ganglia at the anterior end or head of the organism. In addition to the central ganglia, they also have the presence of eye spots, which serve as sensory function. Note that the ganglia will extend into eventually the ventral nerve cords that run along the side of the animal. Also note the presence of a highly branched gastrovascular cavity. This ensures that nutrients are distributed to all cells within the body. Also note that the flattened shape will also help ensure that all cells have access to nutrients, gases, and can undergo the elimination of waste material. Flatworms also have a simple protonephridia, which is a network of tubules that can extend through the body. These tubules will help filter out waste from the main body fluid and also will help in maintaining osmotic balance. The next major group that we will look at are the Syndermata. These are also part of the Lophotrochozoans. Also note that this is a newly revised classification. The phylum Syndermata now represents the rotifers as well as the enkethocephalans. Rotifers are considered to be small microscopic aquatic animals that can be found in marine, freshwater, and damp soils. Rotifers have a rudimentary alimentary canal with a mouth as well as an anus. They also have a ciliated crown that helps to direct food into the mouth area. Some may have the presence of jaws or trophi that can grind up microorganisms as they enter into the digestive tract. In addition, they have a hemocoel that can act as a hydrostatic skeleton. Sexual reproduction can happen in some species, however, many use parthenogenesis to reproduce asexually. The acanthocephalans are considered vertebrate parasites. Many of them lack a complete digestive tract and can reproduce sexually with 2 or more hosts. During their cycle, when they infect a host organism, they typically change its behavior to make it more susceptible to predation, thus increasing its likelihood of locating a secondary host. These organisms are considered to be triploblastic with a hemocoel. They're also considered to be closely related to rotifers, specifically in the Sazon genus. The next 2 major groups of Lophotrochozoans are the ectoprocs and brachiopods. Both of these phyla have a lophophore present in the adult form, a U-shaped alimentary tract with a true coelom. However, they do not have a distinct head region. The ectoprocs are mostly marine, however some species are freshwater. These are also called bryozoans because they have a moss-like appearance similar to the bryozoan plants. Many of these live in sessile colonies and have a hard exoskeleton. However, within the exoskeleton there are pores that the lophophore can extend through, as seen in the diagram to the lower left. Brachiopods, rather, are marine organisms. Many of these are known as lampshell clams. They have a dorsal and ventral side, however they're not considered to be true clams, as we'll see in a few slides. Brachiopods will open their shell to allow water and food to enter into the lophophore for feeding. Another major group of Lophotrochozoans are the mollusks. This is considered to be the second most diverse animal phyla that contains over 100,000 different species. This includes the snails, slugs, oysters, clams, octopuses, as well as squids. Many of these species are marine, freshwater, as well as terrestrial. Most are male and female, however some species, such as snails, are considered to be hermaphroditic. Many of these organisms are soft-bodied, but however some can secrete a shell of calcium carbonate. Their typical body plan includes these major structures: a foot, visceral mass, mantle, mantle cavity, and radula. Take a look at this image here to see the overall structure of a typical mollusk. Note the presence of a mouth, digestive tract, as well as an anus. Also note the presence of a true body cavity or coelom. This slide here depicts the major characteristics of mollusks. These include the chitons, gastropods, bivalves, as well as cephalopods. Finally, the last group of Lophotrochozoans we will discuss are the annelids. These include segmented worms that have a true coelom. These can be found in aquatic and terrestrial habitats and can vary greatly in their length. Annelids were previously grouped into polychaetes, oligochaetes, as well as leeches. However, molecular analysis has proved that this previous grouping was incorrect. Oligochaetes were actually a subgroup of polychaetes, and leeches were a subgroup of oligochaetes. As a result, the newer revision of this system includes two major clades, the Aurantia as well as the Sedentaria. The Aurantia are considered to be mostly mobile and marine. They have well-developed jaws and sensory organs, as you can see in the diagram below. However, a few of the species are immobile and tube-dwelling. Note that each body segment has a pair of parapodia, or fake feet, that are used for locomotion. Parapodia are also seen in other annelids. These parapodia may also be modified to function as gills if the organism is living in an aquatic environment. The sedentarians are considered to be less mobile. Many of these species will burrow into the soil or the sediment and are tube-dwelling. They also typically have pronounced gills or tentacles to help aid in their respiration and feeding, despite the fact that they are immobile. These also include leeches as well as earthworms.